Welcome to Happy Sleep, my curious friends. Have you ever held a leaf up to the sun and wondered, how? How does something so still, so seemingly simple, capture the raw, untamed energy of a star and turn it into the very stuff of life? Or take in a deep breath and in an instant, your brain can tell the difference between the scent of fresh rain and brewing coffee? Or perhaps, in a quieter moment, you've wondered about the very nature of the thoughts you're thinking right now. What are they, and where do they come from? For centuries, we've looked to chemistry and biology for answers. We've broken life down into cells, proteins, and genes. And that works, to a point. But there's a frontier, a deeper layer of reality, where the rules we know seem to melt away. This is the quantum realm. A world where particles can be in two places at once, where information can be teleported, and where an electron can simply vanish from one side of an impossible barrier and reappear on the other. It's a world so bizarre, we assumed it was confined to the freezing cold of physics labs and the hearts of distant stars. But what if we were wrong? What if that bizarre, spooky, counterintuitive quantum world isn't separate from the living world, but its very foundation? Today, we're diving deep into one of the most fascinating and mind-expanding questions in modern science: quantum biology. Does physics govern life's deepest mysteries? We'll explore how the fundamental rules of reality, the physics of the impossibly small, might be the hidden director behind life's most stunning performances, from the near-perfect efficiency of a photosynthesizing plant to the very spark of consciousness in your mind. Before we journey into the quantum undergrowth of life itself, if you're as captivated by mysteries like this as I am, do us a huge favor, smash that like button, and subscribe to join our community of curiosity. This is a wild ride, and I want you with us for all of them. Also, drop a comment below telling us what time you're watching this and where you're joining us from. It's always amazing to see our global family of thinkers gathering together. Now let's begin. Let's start with the process that literally powers our biosphere: photosynthesis. For decades, biologists marveled at its efficiency. A photon of sunlight, a tiny packet of energy, is captured by a vast, sprawling network of chlorophyll antennae in a plant cell. This energy needs to travel to a special reaction center where it's converted into chemical fuel. Think of it like a vast, chaotic stadium, the antenna network, where a single, precious ball of energy, the photon, is caught. It then needs to be passed, flawlessly and instantly, through a jostling crowd of molecules to a specific goal at the other side. Classically, this should be a slow, random walk. It would get lost, dissipated as useless heat. But it isn't. The energy transfer in systems like those of certain bacteria can be over 99% efficient. How? So, we left off with a kind of magic trick. A stadium full of molecules catches one photon of light, and that packet of energy doesn't get lost in the chaotic crowd. Instead, it finds its way to the single, specific exit gate, the reaction center, with over 99% efficiency. And we said the secret isn't classical physics, it's quantum. It's a quantum walk powered by coherence. But what does that really mean? Let's slow this down and get our hands on the nuts and bolts of this quantum shortcut. First, we need to meet the star of the show, the exciton. When a photon smacks into a chlorophyll molecule, it doesn't just heat it up like a sun-warmed rock. It delivers a precise punch of energy that kicks an electron within that molecule into a higher energy state. This creates a bound pair, the excited, energetic electron and the hole it left behind. This duo, dancing together, is the exciton. It's not a physical particle you could hold, it's a quasi-particle, a quantum packet of energy that now belongs to the entire molecular network. Think of it as the hot potato of energy, but a potato that can be in many hands at once. Now, the classical, textbook view of what happens next is what scientists long assumed. This exciton, this hot potato, would randomly hop from one pigment molecule to its nearest neighbor. It would bounce around the chaotic stadium, losing a little energy as useless heat with every single hop, taking a slow, meandering, and frankly, inefficient random walk toward its goal. A lot of potatoes would get dropped along the way. But that's not what happens. The revolutionary discovery, first made in 2007 by Graham Fleming's team at Lawrence Berkeley National Laboratory, changed everything. Using an ultra-fast laser technique called two-dimensional electronic spectroscopy, which is like a super slow-motion camera for quantum events, they saw something astonishing. They didn't see a particle hopping. They saw a wave. For a brief, critical moment measured in femtoseconds, that's millionths of a billionth of a second, the exciton doesn't choose a path. Thanks to the quantum principle of superposition, it behaves as if it's taking all possible paths through the antenna network at the same time. Its quantum wave function spreads out like a ripple in a pond. This is the essence of quantum coherence. The exciton is in a superposition of locations. It's not in one seat in the stadium. It's in a coherent, wave-like pattern across a whole section of the bleachers simultaneously. This wave-like motion is the quantum walk. And it has a superpower, quantum interference. As these probability waves spread out, they overlap. Some paths will interfere constructively. The peaks of the waves line up, making the signal stronger. Others will interfere destructively. Peaks meet troughs and cancel each other out. The system isn't blindly stumbling. It's using wave interference to, in essence, feel out the entire energy landscape at once. It can sense the most efficient route to the reaction center and reinforce that path, while canceling out the dead ends and wasteful detours. It's as if the entire stadium section, holding the energy, could instantly communicate and coordinate to pass the hot potato along the single fastest chain of hands directly to the goal. The signature of this coherent wave-like motion is something beautiful called quantum beating. The Berkeley team observed clear, oscillating signals in their spectroscopic data that lasted for hundreds of femtoseconds. These beats are the direct fingerprint of the exciton's wave nature, like seeing the ripples on a pond interfere with each other. It's the experimental proof that the energy is not localized, but is coherently delocalized across multiple molecules. Now, here comes the biggest head-scratcher of all, the part that really made physicists sit up and take notice. Quantum coherence is famously fragile. We maintain it in our best quantum computers by isolating them in ultra-high vacuums and cooling them to near absolute zero. The interior of a living cell is the absolute opposite of that. It's warm, wet, and wildly noisy, a soup of jiggling molecules and electromagnetic chatter. Physicists assumed any delicate quantum coherence would be instantly obliterated, decohered, in such an environment. It would be like trying to hear a whispered symphony in the middle of a roaring hurricane. Yet the coherence lasts. Not for long in absolute terms, but for just long enough, hundreds of femtoseconds, to complete the energy transfer mission. How? This is where biology's genius engineering shines. It turns out the protein structure that holds the pigment molecules in place isn't just a passive scaffold. It's an active participant. It doesn't just house the chlorophylls, it tunes them. By holding the pigment molecules in exquisitely precise positions and orientations, with what MIT researchers call the quantum Goldilocks effect, the protein creates the perfect conditions for coherent coupling. The distance and angle between molecules is just right to allow their quantum states to talk to each other and merge into a shared, delocalized exciton. Even more fascinating, the noisy cellular environment might not be the enemy of this quantum process. It might be part of the toolkit. Some theories suggest that certain vibrations within the protein itself can actually help to sustain the coherence, or guide the energy transfer. The random jolts from the environment aren't just destructive noise. They can, in a controlled way, give the exciton little nudges that help it explore the energy landscape. It's a partnership between quantum order and classical disorder, a dance at the very quantum/classical border where biology has learned to operate. The protein creates a protected corridor, a quantum corridor as some very recent 2025 research describes it, that shepherds the exciton along high-probability paths while suppressing paths that lead to wasteful decoherence. And where is this all leading? Not just to a deeper understanding of nature, but to a new technological frontier. At MIT, a team led by Professors Angela Belcher and Seth Lloyd asked a brilliant question: if nature uses precise molecular architecture to enable quantum-enhanced energy transport, can we copy it? Their answer was to hijack a biological blueprint. They genetically engineered a virus, a harmless one used in biotech, to act as a scaffold. They then decorated this virus with synthetic light-absorbing dye molecules, tweaking the virus's genes to space these dyes at the perfect Goldilocks distance. And it worked. They created a synthetic, virus-based light harvester that demonstrated enhanced energy transport through quantum coherence, more than doubling the speed and distance the exciton could travel. This is more than proof of concept, it's a roadmap. It shows us that by learning the quantum architectural secrets of a leaf, we might one day build solar cells, sensors, and computers that are as efficient and elegant as nature's own designs. So, we've just witnessed a miracle of energy transport. We saw how a leaf uses quantum weirdness as a superhighway to move sunlight's energy. Now, let's step inside the leaf, inside the cell itself, and meet the true workforce, the enzymes. If the quantum coherent antenna network is the dazzling, high-speed interstate, then enzymes are the bustling, impossibly efficient factories that line its exits. They take the delivered raw energy and raw materials and perform the precise, molecular-scale craftsmanship that builds and powers life itself. And as we're about to see, their most essential tool isn't just a chemical wrench, it's a quantum ghost. Think of your body right now. You're breathing, your heart is beating, your brain is deciphering these words. Every single one of those processes is powered by a continuous, invisible cascade of chemical reactions. If you had to rely on the raw, unassisted speed of these reactions happening in a test tube, life would grind to a halt. They'd be far too slow. We need these reactions to happen millions, even billions of times faster. Enter the enzyme. Enzymes are biological catalysts. They are typically protein molecules, though some are made of RNA, that have one magnificent job: to accelerate a specific chemical reaction without themselves being used up in the process. They are the master craftsmen, the specialized artisans of the cellular world. One enzyme might be exquisitely designed to stitch two molecules together. Another, like a pair of molecular scissors, might be designed to chop one apart. And their power of acceleration is not modest. We're talking about making reactions proceed 10 to power 3 to 10 to power 17 times faster than they otherwise would. Without that speed, there is no life. Now, how does a humble-looking protein molecule perform such magic? For a long time, the textbook explanation was elegant and mechanical. Imagine an enzyme as a specially shaped lock. The molecule it acts upon, called the substrate, is the key. The substrate slips into a precisely molded pocket on the enzyme called the active site. Once snugly bound, the enzyme might stretch a particular bond in the substrate, making it easier to break, or it might hold two substrates in the perfect orientation for them to connect. In doing so, it lowers the activation energy, the initial push needed to get the reaction started, and provides a safer, more efficient pathway from reactants to products. This model, known as Michaelis-Menten kinetics, describes this dance beautifully. Enzyme, E plus substrate, S, forms a quick complex, ES, which then transforms to release the product, P, and the free enzyme, ready to go again. The speed of this process hits a maximum, called Vmax, when every single enzyme molecule is busy, saturated with its own substrate. This is the classical view. And it works. It explains so much. But as we peer deeper into the very heart of the reaction at the scale of single atoms, we find puzzles. Some reactions that enzymes catalyze are so fast, so efficient, that just lowering the activation energy barrier doesn't seem to fully account for it. It's as if the craftsman isn't just providing better tools, he's occasionally walking through the workbench itself. This brings us to the star of our quantum show within the enzyme, the humble proton. A hydrogen ion. The smallest, lightest, and most nimble thing an enzyme is often tasked with moving. In countless fundamental reactions, the very ones that drive your metabolism, that help extract energy from food, that copy your DNA, a critical step is the transfer of a single proton from one part of a molecule to another. It's the ultimate game of molecular hot potato. And here lies the classical problem, the locked room with no door. For the proton to get from the donor molecule to the acceptor molecule, it must overcome an energy barrier. Think of it as a hill it must climb. On one side of the hill is the reactant state, the substrate with the proton attached. On the other side is the product state, the substrate having donated the proton. The peak of the hill is the high-energy, unstable transition state. According to classical physics, which views particles like tiny billiard balls, the proton must gather enough energy, the activation energy, to roll over the top of that hill. If it doesn't have the energy, it stays put. The enzyme's job, classically, is to lower that hill, making it easier to climb. But sometimes, the hill is still too steep, or the reaction needs to be faster than even a lowered hill allows. This is where the quantum world whispers its solution. In the quantum realm, particles like protons are not just balls. They have a dual nature. They are also waves. This isn't a metaphor, it's a fundamental property of reality. And a wave facing a barrier behaves very differently than a ball does. Imagine you're standing in front of a thick, solid wall, holding a basketball. If you throw the ball at the wall, it bounces back. Always. Now imagine you are a sound wave. You approach the same wall. Some of you will reflect back, yes. But some of your energy, some of your essence, will travel through the wall as vibration and be heard faintly on the other side. The wall, to a wave, is not an absolute, impenetrable border. It is a filter. This is the essence of quantum tunneling. The proton, behaving as a quantum wave, has a probability, not a certainty, but a probability, of simply appearing on the other side of the energy barrier without ever having existed at the top. It doesn't jump. It doesn't climb. From the perspective of the wave function describing its location, it fades from one stable location, exists fleetingly as a probability cloud that extends through the barrier, and coalesces into a particle on the other side. The wall, for a quantum particle, has a finite thinness. For a proton facing a narrow enough barrier, tunneling from donor to acceptor isn't a miraculous exception, it's a regular, probabilistic event. This changes everything for our enzyme. It's no longer just a machine that lowers hills. It is an architect that designs tunnels. The reaction no longer depends solely on the proton having enough thermal energy to surmount a barrier. It can now also proceed via the proton quantum mechanically tunneling through a barrier. At the temperatures of life, for the right kind of narrow barrier, tunneling can be the dominant, faster pathway. But nature, in its boundless sophistication, doesn't just leave this to chance. Evolution has sculpted enzymes to be master engineers of the quantum realm. They don't passively hope for a tunneling event, they actively promote and optimize it. How? First, by exquisite positioning. The enzyme's active site grips the donor and acceptor molecules with atomic precision. It holds them at a perfect, fixed distance and orientation relative to each other. Why is this so critical for tunneling? Because the probability of tunneling doesn't just depend on the height of the energy barrier, how steep the hill is, but even more dramatically on its width, how thick the wall is. The tunneling probability drops exponentially with increasing barrier width. By holding the reactants in a tight, perfect embrace, the enzyme ensures the proton has the shortest possible path to tunnel. It's not just bringing the two sides of the chasm closer, it's pouring concrete to create the narrowest possible tunnel. Second, by environmental tuning. The interior of an enzyme's active site isn't a passive hollow. It's a dynamic, chemically rich environment. Specific amino acid residues create precise electrical fields. These fields can subtly reshape the energy landscape of the reaction. An enzyme can tweak the electrical environment to not just lower the barrier, but to make it thinner in a specific spot, creating an even more favorable tunnel for the proton. Furthermore, enzymes aren't static statues. They breathe. They have internal vibrations and movements. Remarkably, research suggests that certain coordinated vibrations of the enzyme's own structure can momentarily align to create a transient, even more favorable corridor for tunneling, a process called vibrationally enhanced tunneling. The enzyme is essentially using its own quantum mechanical jiggles to shake the proton through the barrier at the optimal moment. This leads us to one of the most profound implications. Quantum tunneling helps explain the breathtaking specificity of enzymes. An enzyme must not only be fast, it must be exquisitely choosy, reacting with its correct substrate and almost nothing else. The classical lock and key model explains some of this. But tunneling adds a quantum-honed edge. For the wrong substrate, even if it fits in the active site, the precise alignment for efficient tunneling won't be there. The donor and acceptor orbitals, the quantum doors the proton must tunnel between, won't line up correctly. The barrier width will be just a tiny bit too large, causing the tunneling probability to plummet. Thus, the enzyme's quantum design ensures that only the perfect substrate gets the full benefit of the quantum shortcut. It's a built-in, subatomic proofreading mechanism. We can now see the full, beautiful picture. An enzyme is a quantum-enabled machine. It uses its classical structure, its uniquely folded protein shape, to create a quantum mechanically optimized chamber. It positions atoms with nanometer precision to define the shortest tunneling path. It uses electric fields and its own dynamics to fine-tune the quantum landscape. And in doing so, it harnesses the inherent wave-like probability of the proton to achieve reaction speeds that are the very foundation of a living, breathing, thinking organism. The proton transfer in your cells, powering your every thought and motion, isn't just a chemical event. It's a quantum event. Life isn't just using chemistry. At its core, in its most fundamental act of transformation, it is leveraging the strange, probabilistic rules of the quantum universe. The enzyme isn't just a craftsman with great tools. It is a guide, leading a proton through a ghostly mountain pass that, according to the old rules of physics, shouldn't even exist. And it does this billions of times a second, in every cell in your body, right now. That is the silent, quantum heartbeat of life itself. So, we've journeyed through the quantum green of the leaf and into the molecular tunnels of the enzyme. Now, let's lift our gaze. Let's follow a traveler on one of the most epic and mysterious journeys on our planet. Picture a European robin, a small, plump bird you might see in a garden, weighing little more than a few coins. As autumn frosts begin to bite in Scandinavia, this tiny creature feels an ancient urge. It takes to the night sky, embarking alone on a journey of thousands of kilometers over featureless sea and unfamiliar land, aiming with uncanny precision for a specific cluster of trees in North Africa where it will spend the winter. And in the spring, it will do it all again in reverse. This raises a profound question, one that has puzzled humans for centuries. How? How does it know? It's not following a parent, young birds on their first migration are following an inherited genetic vector, a direction and duration programmed into them. They have no map, no experience. They use the sun and the stars as compasses, yes, learning to calibrate them with an internal clock. But on cloudy nights, or under the dense canopy of a forest, those celestial cues vanish. Yet the journey continues. The birds clearly have another guide, one that is always present, day or night, clear or cloudy, the Earth's magnetic field. For decades, the simplest assumption was that birds must have a kind of biological compass needle. And indeed, scientists found tiny deposits of an iron-rich mineral called magnetite in the beaks of pigeons and other birds. It seemed like a perfect answer, a physical, mechanical compass. But as they probed deeper, problems arose. The primary compass used by migratory songbirds like the robin doesn't behave like a polarity compass, one that points to north and south. It's an inclination compass. It senses the angle at which the magnetic field lines dive into the earth. To a robin, the important information isn't magnetic north, it's toward the pole or toward the equator, which it senses by whether the field lines are tilting up or down. A simple magnetite needle would be flipped 180 degrees if you reverse the field's polarity, but a bird's compass wouldn't be. Furthermore, this magnetic sense is light-dependent. It only works in the presence of blue to green light. You can disrupt a bird's orientation not with a strong magnet, but with a very weak, rapidly oscillating radio frequency field, which would do nothing to a chunk of magnetite. These bizarre properties pointed away from a mechanical solution and towards something far more exotic. The evidence began to suggest that birds don't just sense the magnetic field. They might literally see it. This brings us to the radical pair mechanism, a theory so elegant and strange it sounds like science fiction. It proposes that the bird's magnetic compass is a quantum sensor, built from light and spun from the most delicate thread of quantum coherence. And it all starts with a single photon of blue light. Deep within the retina of a migratory bird's eye is a special protein called cryptochrome. In birds, a specific variant called cryptochrome 4, or Cry4a, is particularly interesting. This protein acts like a tiny, tuned antenna. When a blue photon strikes its core, it excites a molecule called FAD, flavin adenine dinucleotide, giving it a burst of energy. This energy triggers a game of molecular hot potato. An electron is stripped from a nearby amino acid called tryptophan and handed to the excited FAD. In an instant, this creates a precarious situation. The tryptophan is now missing an electron, making it a radical, and the FAD has an extra one, making it another radical. Together, they form a radical pair, two molecules, each with an unpaired electron, created at the same moment. Now, electrons are not just tiny charges. They possess a quantum property called spin. Think of spin not as literal spinning, but as an intrinsic magnetic orientation, like a microscopic arrow or a tiny compass needle pointing in a specific direction. When the radical pair is born, the spins of these two unpaired electrons are not independent. They are intimately linked, or quantum entangled. Their fates are joined. This entangled pair exists in a superposition of two possible collective states. In the singlet state, the two electron spins are opposite, antiparallel. In the triplet state, they are aligned parallel. Immediately after creation, this linked system doesn't settle on one state. It begins a rapid, delicate quantum waltz, oscillating between the singlet and triplet states millions of times per microsecond. This oscillation is the heartbeat of the quantum compass. Here is where the impossibly weak whisper of the Earth's magnetic field, about 100 times weaker than a common refrigerator magnet, makes its entrance. This gentle field influences the precession of the electron spins. Precession is like the wobble of a spinning top. The axis of the spin itself slowly rotates. The Earth's magnetic field subtly changes the rate of this wobble for each electron. And because the two electrons are entangled, the field's effect on one instantly influences the state of the other. It's as if you had two interconnected spinning tops on opposite sides of a table. Nudging one would instantly change the wobble of the other. This external magnetic nudge interferes with the innate quantum waltz of the radical pair, changing the probability of whether the pair ends up in a singlet or triplet state after a few microseconds. This quantum mechanical outcome, singlet or triplet, is the key. It determines the ultimate chemical fate of the cryptochrome protein. Depending on which state the radical pair is in when the electron jumps back to its original home, the protein can be left in a slightly different chemical form. One form might be active, the other inactive. The probability of forming the active form depends on the angle between the cryptochrome molecule and the Earth's magnetic field lines. Imagine now not one, but millions of cryptochrome molecules, fixed in a sheet across the bird's retina, like pixels in a screen. Each one is oriented in a slightly different direction. As the bird turns its head, the angle between each cryptochrome pixel and the magnetic field changes. So, across the retina, a pattern of activity emerges. Cryptochromes aligned in one direction might be more likely to become active, while those aligned another way might not. This creates a visual pattern, a dim, shadowy overlay on the bird's normal sight. It might be a gradient of darkness, a blotch, or a compass bearing painted directly onto the world. A neuroscientist studying European robins found that a specific brain region, cluster N, which processes visual information, lights up with activity when the birds use their magnetic compass, powerfully linking this sense to vision. The evidence for this quantum dance is compelling. Researchers extracted the gene for the European robin's cryptochrome, produced the protein in the lab, and demonstrated it is exquisitely sensitive to magnetic fields, much more so than the same protein from non-migratory chickens. The protein's sensitivity peaks during migration season. In behavioral experiments, a weak radio frequency field, tuned to the exact frequency of the singlet-triplet oscillation, completely scrambles a robin's orientation because it drowns out the subtle quantum signal. Even more telling, when scientists in one lab couldn't get their robins to orient at all, they discovered the cause was imperceptible radio noise from nearby electronic equipment on their campus. When they shielded their wooden huts with grounded aluminum, which blocks radio waves but not magnetic fields, the bird's quantum compass started working perfectly again. It's important to note that the story may be even richer. Many scientists believe birds use a multi-system approach. The radical pair mechanism in the eye provides the primary directional compass. The magnetite in the beak might act as a crude intensity sensor, part of a map sense that helps them roughly gauge their position based on variations in the Earth's magnetic field strength. And very recent, groundbreaking research from 2025 has identified a potential third system in the inner ear, where specialized hair cells may detect magnetic fields through electromagnetic induction, possibly helping with balance and 3D orientation. The radical pair mechanism, however, remains the leading and most well-supported explanation for the precision compass that guides those epic, starlit flights. So, the next time you see a robin in your garden, or hear of a godwit flying non-stop from Alaska to New Zealand, consider the wonder happening behind its eyes. It is not just looking at the world. It is perceiving an invisible grid of force that blankets our planet, using a sensor built from the most counterintuitive rules of reality. It is converting the silent whisper of a global magnet into a visual cue through a chemical reaction steered by quantum entanglement. In the dark, guided by starlight and this quantum overlay, a tiny bird is holding in its mind a map woven from the fabric of physics itself, following a path written in light and spin toward a home it has never seen. So, we've followed the quantum shortcut through the leaf, marveled at the proton's ghostly tunnel in the enzyme, and even glimpsed the magnetic compass painted in quantum spin behind a bird's eye. Now, let's bring this wonder home. Let's turn the quantum lens on one of our most intimate, evocative, and mysterious senses, smell. Close your eyes for a moment. Think of the smell of freshly cut grass, of rain on hot pavement, of your grandmother's kitchen, or the pages of an old book. In an instant, these scents can transport us across time and space, triggering memories and emotions with a power that vision or sound rarely matches. But how does this happen? How does a physical molecule, drifting through the air, become a feeling, a memory, a perception inside your mind? For over a century, the dominant answer has been elegant and mechanical: the lock and key theory. Imagine an odorant molecule, the tiny key. It floats into the olfactory epithelium in your nose, a patch of tissue covered in millions of olfactory receptor neurons. At the tips of these neurons are the LOCs, olfactory receptor or proteins. According to the theory, if the shape of the odorant key fits snugly into the pocket of the receptor lock, it turns, triggering a signal that races to your brain: grass, or coffee, or rose. It's a beautifully intuitive idea, explaining why we might need hundreds of different receptor types to detect thousands of smells in a combinatorial code. And indeed, some receptors do show a degree of shape specificity. But, as with so much in biology, the closer you look, the messier and more wonderful it gets. The lock and key model runs into some perplexing mysteries that hint at a deeper layer of reality. The first mystery is that molecules with drastically different shapes can smell remarkably similar. Consider the classic example of musk. Natural musk from the musk deer has a complex, ring-heavy structure. Yet chemists have created synthetic musks, like galaxolide, that share almost no structural resemblance to the natural compound, but whose scent is virtually indistinguishable to us. If smell is purely shape-based, how can two completely different keys open the same lock? The second, and perhaps more profound, mystery involves identical shapes with different weights. This is where isotopes come in. An isotope is a version of an atom that has the same number of protons, defining its chemical identity, but a different number of neutrons, making it heavier. You can replace, say, a hydrogen atom, the lightest element, in an odorant molecule with its heavier isotope, deuterium. Crucially, this substitution does not change the molecule's shape. The bonds are the same length, the angles are the same. From a lock and key perspective, it is the exact same key. Yet, in numerous experiments, organisms from fruit flies to humans can sometimes tell the difference. A heavier molecule can smell subtly, but distinctly different. This is a massive clue. If the physical shape is identical, but the smell changes, then the receptor must be detecting something other than shape. It must be sensing some intrinsic property of the molecule that changes when you add that tiny bit of nuclear weight. And this is where our journey takes a quantum leap. Enter the vibration theory of olfaction, sometimes called the quantum nose hypothesis. First proposed by Malcolm Dyson in the 1930s and refined by Luca Turin in the 1990s, this theory suggests that what we smell is not a molecule's shape, but its vibrational song. To understand this, we need to think of a molecule not as a static tinker model, but as a dynamic, vibrating entity. The bonds between atoms, those little sticks in the diagram, aren't rigid. They're more like springs. And just like a guitar string, they can vibrate at specific, quantized frequencies. Stretch a carbon-hydrogen bond, and it plucks at a certain frequency. A carbon-deuterium bond, being heavier, vibrates more slowly, at a lower frequency. A carbon-oxygen double bond has its own, higher-pitched note. Every molecule has a unique spectrum of these vibrations, a quantum mechanical fingerprint determined by the masses of its atoms and the stiffness of its bonds. Biological spectroscope, a tiny machine designed to listen to this molecular song. But how could a protein possibly detect such a thing? This is where our old friend quantum tunneling returns for an encore, but in a spectacular new role. The theory suggests the mechanism is inelastic electron tunneling. Let's set the stage. Imagine an odorant molecule has docked in the pocket of an olfactory receptor. Within the protein, there are two sites that an electron would love to jump between, like two stepping stones across a stream. The only problem is a small energy barrier between them. Normally, the electron doesn't have the energy to hop across. Here's the quantum magic. Thanks to its wave-like nature, the electron can tunnel through this barrier. This is elastic tunneling. It goes through without losing energy, like a ghost passing through a wall. But there's another possibility. As the electron tunnels, it can lose a precise, tiny packet of energy. This is inelastic electron tunneling. It's as if the electron has to pay a toll to get through the barrier. Where does that energy toll go? According to the theory, it is donated to the odorant molecule sitting in the receptor's pocket. If the energy packet exactly matches the energy needed to excite one of the molecule's specific vibrational modes, say, to get that carbon-oxygen bond swinging, the energy is absorbed. The molecule begins to vibrate in that specific way. The electron's successful, inelastic tunnel is therefore dependent on the presence of a vibration at just the right frequency. The receptor protein is exquisitely tuned to detect this event. The vibration of the odorant molecule, now energized, could nudge the protein, catalyzing a subtle change in its own shape or electrical state, ultimately triggering the neural signal. I detected a vibration at this frequency. Think of it this way: the receptor provides a gate for an electron. The odorant molecule provides the keyhole for that gate, but the keyhole only opens if you sing the right note. The molecule's vibrational frequency is the secret melody that unlocks the sense of smell. It's not about the physical shape of the key, it's about the quantum mechanical song it sings when the electron flows. This elegant idea elegantly solves our two mysteries. Why do different shapes smell the same? Because they might share a similar vibrational fingerprint. Two molecules with wildly different architecture could have bonds that vibrate at similar sets of frequencies, singing a similar song to the receptor. Why do isotopes smell different? Because swapping hydrogen for deuterium changes the vibrational frequency of those bonds. A carbon-hydrogen bond and a CD bond sing different notes. The same shaped key now hums a lower pitch, and the receptor tuned to the higher pitch might not recognize it, while another might. The most compelling, direct evidence for this theory comes not from humans, whose perceptions are subjective and culturally influenced, but from the humble fruit fly, Drosophila melanogaster. In a landmark 2011 study, researchers presented flies with a simple choice. They used odorants like acetophenone, which has a sweet, orange blossom smell to us, and created versions where hydrogen atoms were replaced with deuterium. To a shape-only model, these should be identical. The flies begged to differ. They could clearly, consistently tell the normal and deuterated versions apart. Even more brilliantly, the researchers then trained the flies. They taught one group to associate an electric shock with the normal acetophenone, and another group to associate the shock with the deuterated version. Then they tested them with a completely different pair of odorants, like normal and deuterated benzaldehyde, which smells of almonds. Astonishingly, the flies generalized their learning. Flies trained to avoid the deuterated acetophenone also avoided the deuterated benzaldehyde. Flies trained to avoid the normal acetophenone avoided the normal benzaldehyde. This is a knockout result for the vibration theory. The flies weren't recognizing a specific shape or chemical group. They were recognizing the abstract property of deuteratedness, the common, lower-frequency vibrational signature imparted by the heavier deuterium atoms. They were smelling the quantum vibrational song. Furthermore, structural biology has shown that olfactory receptors are often broadly tuned and promiscuous. They recognize patterns of physico-chemical properties, like hydrophobicity or polarity, more than ultra-specific shapes. A 2021 study of an insect receptor, MHOR5, revealed it has a single, geometrically simple binding pocket that recognizes diverse odorants through distributed, non-specific hydrophobic interactions. This seems to support a model based on general chemical feel rather than a precise vibrational spectroscope. However, vibration theorists might counter that this very promiscuity is what you need to allow many different shaped molecules that share a vibrational profile into the same pocket to be listened to. The truth, as it so often is in biology, is likely to be a magnificent, messy hybrid. Our sense of smell is almost certainly combinatorial and multifactorial. The physical shape and chemical character of a molecule undoubtedly matter for how it initially docks and interacts with the receptor protein. This docking event is the first step. But once docked, the final act of triggering the receptor, the precise mechanism of signal transduction, could very well involve a quantum vibrational check. It could be a two-step process, a classical handshake followed by a quantum password verification. This is the breathtaking implication of the quantum nose. It suggests that quantum tunneling, the same process that lets protons ghost through enzyme barriers, might also form the basis of a sophisticated sensory code. It transforms our understanding of smell from a simple mechanical fitting to a form of biological spectroscopy. We are not just matching shapes. We are, at some fundamental level, listening to the quantum harmonies of the molecular world. The scent of a rose, the tang of a lemon, the musk of the earth after rain, these are not just chemical signals. They are symphonies of vibrating bonds, translated into perception by a biological system sensitive enough to hear the music of the atoms themselves. So, the next time you catch a fragrance on the breeze, pause. Consider that you might not just be detecting a molecule's form. You might be tuning in, with a quantum receiver built by millions of years of evolution, to the unique, vibrational song it sings to the universe. In that fleeting scent, you are connecting with the deepest, most fundamental vibrations of reality. And now, we arrive at the most profound and speculative frontier of all, consciousness. This is the final frontier, not of space, but of inner space. And the question we're tackling isn't just a scientific how, it's a profound why. This is what philosopher David Chalmers famously dubbed the hard problem of consciousness. Let's break down why it's so famously hard. Science is spectacularly good at explaining how things work. These are the so-called easy problems, though. As cognitive scientist Steven Pinker notes, they're only easy in comparison, more like curing cancer than fixing a toaster. How do our eyes take in light and turn it into signals? How do those signals get processed in the visual cortex? How does that processing lead us to duck when we see a ball flying at our head? Neuroscience is making incredible strides on these functional questions. Let's break down why it's so famously hard. Science is spectacularly good at explaining how things work. These are the so-called easy problems, though. As cognitive scientist Steven Pinker notes, they're only easy in comparison, more like curing cancer than fixing a toaster. How do our eyes take in light and turn it into signals? How do those signals get processed in the visual cortex? How does that processing lead us to duck when we see a ball flying at our head? Neuroscience is making incredible strides on these functional questions. But the hard problem asks something else entirely. It asks, why is all that physical, electrochemical information processing accompanied by an inner, subjective experience? When those specific wavelengths of light hit your eye and that cascade of neurofiring happens, why does it feel like the color red? Why does the complex neural pattern associated with tissue damage feel like the raw, aversive quality of pain? You could, in principle, imagine a perfect robot, a philosophical zombie, that processes all the same information, dodges the ball, says ouch. but has no inner movie, no lights on inside. The hard problem is the question of why we are not zombies. It's the problem of qualia, the felt, qualitative what-it's-likeness of every moment of our lives. The philosopher Thomas Nagel captured this perfectly in his essay What Is It Like to Be a Bat? We could know everything objective about bat biology, their echolocation, their neurology, their evolutionary history. But that objective, third-person knowledge does not, and seemingly cannot, grant us the subjective, first-person knowledge of what it feels like to navigate the world through sonar. There is an explanatory gap between the physical description of the brain and the phenomenal reality of the mind. This gap suggests to some that our usual tools of science might hit a limit here, or that we need to consider something fundamentally different. This is where quantum physics enters the debate, not as a magic wand, but as a potential source of new concepts. If the classical picture of the brain as a wet computer made of biochemical switches seems to leave out the lights-on quality, could the bizarre, non-local, fuzzy rules of the quantum realm provide the missing ingredient? One of the most famous and controversial attempts to answer yes is the orchestrated objective reduction theory, proposed by mathematical physicist Sir Roger Penrose and anesthesiologist Stuart Hameroff. Their theory is a breathtaking two-part cosmic ballet. Part 1 comes from Penrose. He was deeply influenced by Gödel's incompleteness theorems in mathematics, which hint that human understanding reaches beyond mere algorithmic computation. Penrose speculated that a new type of physics, a bridge between quantum mechanics and gravity, might be involved in consciousness. He proposed that in tiny structures in the brain, quantum superpositions, those states of being in multiple places at once, naturally grow unstable and collapse into a single reality. But this isn't the random collapse of textbook quantum mechanics. Penrose called it objective reduction, a self-collapse driven by the very mass of the superposition system warping spacetime. Now, what are these tiny structures in the brain where this quantum magic might happen? This is Hameroff's contribution. He pointed to microtubules. These aren't neurons, they're the internal scaffolding, the cytoskeleton, inside every neuron and other cells. Imagine them as tiny, hollow, honeycomb tubes that give the cell its shape and act as information highways. Hameroff proposed that the precise, lattice-like structure of microtubules could support and protect quantum superpositions in proteins called tubulin. The theory suggests these tubulin proteins can act like biological qubits, the basic units of quantum information. Here's the grand fusion. Penrose's objective reduction provides the orchestral score, the fundamental reason for collapse. Hameroff's microtubules provide the instruments, the physical site in the brain. The orchestration comes from the brain's own biological inputs, our senses, memories, and neural activity. They tune the quantum vibrations in the microtubules, influencing how the superposition collapses. And in the orchestroar model, each of these orchestrated, non-random collapses is a tiny, discrete moment of proto-consciousness. The smooth stream of our awareness, then, is the integrated symphony of billions of these quantum events happening across vast networks of neurons. It's a magnificent, all-encompassing vision. But from the moment it was proposed, it faced a thunderstorm of skepticism. The central objection is the warm, wet, and noisy problem. Quantum states like coherence and superposition are notoriously delicate. We maintain them in quantum computers by isolating them in ultra-high vacuums and cooling them to near absolute zero. The interior of a living brain is the polar opposite. It's at body temperature, awash in salty fluids, and buzzing with electromagnetic noise from all the other biochemical activity. Most physicists argue any meaningful quantum coherence in microtubules would be destroyed by this environment in femtoseconds, quadrillionths of a second, far too quickly to play any role in thought processes that happen on the scale of milliseconds. Skeptics, including many neuroscientists, also argue that classical explanations of neural networks are sufficient. They point to complex patterns of brainwave oscillations, where billions of neurons fire in coordinated rhythms, as a more plausible candidate for the neural correlate of consciousness. As neuroscientist Jennifer Parasini explains, electrical waves and neuronal firing should be seen as complementary, but not competing frameworks. In this classical view, consciousness is an emergent property of vast, complex neural computation, with no need for quantum spookiness. Yet, intriguing clues persist that keep the quantum hypothesis alive. Some of the most tantalizing come from the world of anesthetics, Stuart Hameroff's own field. Experiments have shown that different isotopes of the gas xenon have different potencies for knocking out consciousness. Isotopes are atoms that are chemically identical, same number of protons and electrons, but have different masses due to extra neutrons. For example, xenon-129 has a different nuclear spin, a quantum property, than xenon-132. If consciousness were purely a classical, chemical process, these nearly identical gases should work the same. The fact that they don't suggests the brain's switch for consciousness might be sensitive to these subtle quantum properties. Other theories flip the orchestrated objective reduction theory script entirely. Christoph Koch, a leading neuroscientist who has collaborated with quantum physicists, has proposed that consciousness might not correlate with the collapse of quantum superpositions, but with their formation. In this view, a system with a more complex, more widespread, more robust superposition might have a richer conscious experience. Consciousness, then, wouldn't be in the discrete clicks of collapse, but in the expansive, fuzzy cloud of quantum possibility. This idea dovetails with other modern theories, like integrated information theory, which seeks to quantify consciousness in terms of the interconnectedness of a system, a property quantum entanglement could, in principle, amplify exponentially. This brings us to perhaps the most mind-bending experimental concept on the horizon, championed by researchers like Hartmut Neven of Google's Quantum AI Lab. Neven suggests we stop just theorizing and actually test the quantum consciousness hypothesis by doing something straight out of science fiction, creating quantum entanglement between a human brain and a quantum computer. Think of it as an expansion protocol. The idea is to use gentle, non-invasive techniques, perhaps based on nuclear magnetic resonance, to entangle the quantum states of particles in a subject's brain with the qubits in a quantum processor. You're essentially creating one larger, unified quantum system that spans biology and silicon. The hypothesis is that by expanding the brain's quantum system in this way, you might temporarily alter or enhance the subject's conscious experience. They might report a change in the quality, depth, or unity of their awareness. It's an audacious proposal, and the technical and ethical hurdles are immense. But it frames consciousness in a radical new light, not as a passive byproduct of classical computation, but as an active phenomenon that might be fundamentally tied to the quantum informational structure of the universe. Some, like physicist Michael Pravica, even take this further, suggesting we might be best understood as complex interference holograms, patterns of waves where our conscious sense of self is the unified harmony that emerges. This leads to the most profound shift of all. What if we have it backwards? What if consciousness isn't produced by the brain? This is the foundation priority view. Pioneering quantum physicists like Erwin Schrödinger hinted at it, and modern researchers like Professor Maria Strom of Uppsala University are giving it new mathematical form. In this model, consciousness is the fundamental field of reality, the ground of being. Time, space, matter, and individual brains like ours are then derivative. They crystallize or precipitate out of this primordial conscious field. In this view, the brain isn't a generator of light, it's a receiver, a transducer, tuning into a consciousness that was already everywhere. So, where does this leave us? On a razor's edge between science, philosophy, and profound mystery. The orchestra model, for all its controversy, forced a crucial question: could the brain be more than a classical computer? The clues from anesthetics and the wild proposals for brain quantum entanglement keep that question wide open. Whether the answer lies in microtubules, in brain-wide quantum fields, or in a consciousness that precedes the brain entirely, one thing is clear. The investigation into quantum consciousness is more than a niche scientific debate. It's the ultimate expression of our need to understand our own existence. It challenges our materialist assumptions, pushes the boundaries of testable science, and forces us to confront the possibility that the intimate, vivid reality of our inner lives might be connected to the deepest, most fundamental fabric of the cosmos. The hard problem remains, but in the quest to solve it, we are exploring not just the brain, but the very nature of reality itself. So, where is all this headed? The future of quantum biology is about moving from observing mysteries to building mastery. First, quantum-enabled medicine and sensing. If cellular communication, magnetic field sensing, and enzyme catalysis rely on quantum spin states, then we can potentially manipulate them with tailored magnetic fields. Imagine non-invasive devices that use specific electromagnetic frequencies to steer stem cells to repair tissue, boost mitochondrial function, or even target cancer cells with exquisite precision. We could have a quantum codebook for physiology. Second, bio-inspired quantum technology. Nature has spent eons perfecting quantum tricks. By reverse engineering photosynthesis, we could build ultra-efficient, organic solar cells. By understanding bird navigation, we could develop room temperature, exquisitely sensitive quantum magnetometers for everything from medical imaging to geological surveying. Finally, it forces a philosophical and scientific revolution. Quantum biology bridges the hardest sciences of the very small and the complex sciences of the very living. It demands a new kind of scientist and a new model of collaboration. It suggests that to truly understand life, from the migration of a bird to the feeling of love, we cannot stop at the molecular. We must dive into the subatomic, quantum layer of reality where matter and information, particle and wave, are inextricably fused. So, does physics govern life's deepest mysteries? The evidence is building that the answer is a resounding, awe-inspiring yes, but not the classical physics of billiard balls and levers. It's the strange, wondrous, and profoundly beautiful physics of the quantum realm. Life is not fighting against the weirdness of the quantum world. It is leaning into it, dancing with it, harnessing superposition, entanglement, and tunneling to perform the miracles of sensing, navigating, transforming energy, and perhaps even experiencing. We are not just biochemical machines. We are quantum biological beings, living at the exquisite edge where the cosmic rules of the very small orchestrate the grand, vibrant symphony of the living world. This is more than just a new field of science. It's a new way of seeing our place in the universe. The quantum magic isn't just out there in the void. It's right here, in the green of the leaves, in the song of a migrating bird, in the scent of a rose, and in the silent, luminous theater of your own mind. Thank you so much for joining me on this incredible journey to the quantum heart of life. If your mind is buzzing with as many questions and wonders as mine is, let's keep the conversation going. Hit that like button if you enjoyed the dive. Subscribe so you don't miss our next exploration into the mysteries of science and existence. And most importantly, tell me in the comments, what part of this quantum biological world blew your mind the most? Was it the bird's quantum compass? The idea of a quantum smell? Or the wild possibility of a quantum mind? Until next time, stay curious, look a little deeper at the world around you, and remember, the universe is far stranger and more wonderful than we ever imagined. Goodbye for now.